Topic of today's lecture is nucleic acids, plan, definitions, DNA, RNA, structural levels of nucleic acids. Nucleic acids are biopolymers. The monomers are called nucleotides, and each consists of three components: a nitrogenous heterocyclic base, either purine or pyrimidine, a pentose sugar, and a phosphate group. Different nucleic acid types differ in the specific sugar found in their chain. Also, the nitrogenous bases possible in the two nucleic acids are different. Adenine, cytosine, and guanine are possible in both RNA and DNA, while thymine is possible only in DNA and uracil is possible only in RNA. So nucleic acids are composed of nucleotide monomers linked together. The nucleotides contain three parts: a nitrogenous base, a 5-carbon sugar, a phosphate group. Nucleotides are linked together to form polynucleotide chains. Nucleotides are joined to one another by covalent bonds between the phosphate of one and the sugar of another. These linkages are called phosphodiester linkages. Phosphodiester linkages form the sugar-phosphate backbone on the both DNA and RNA. Similar to what happened with protein and carbohydrate monomers, nucleotides are linked together through dehydration synthesis. In nucleic acid dehydration synthesis, nitrogenous bases are joined together and the water molecule is lost in the process. Interestingly, some nucleotides perform important cellular function as individual molecules, the most common example being ATP. Nucleic acids, DNA. DNA is the cellular molecule that contains instructions for the performance of all cell functions. When a cell divides, its DNA is copied and passed from one cell generation to the next generation. It is the main function of DNA, transmitting the genetic information. DNA is organized into chromosomes and found within the nucleus or our cells. It contains the programmatic instructions for cellular activities. When organisms produce offspring, these instructions are passed down through DNA. DNA commonly exists as a double-stranded molecule with a twisted double helix shape. DNA is composed of a phosphate deoxyribose sugar backbone and the 4 nitrogenous bases: adenine A, guanine G, cytosine C, and thymine T. In double-stranded DNA, adenine pairs with thymine AT and guanine pairs with cytosine G, C. Nucleic acids, RNA. RNA is essential for the synthesis of proteins. Information containing within the genetic code is typically passed from DNA to RNA to the resulting proteins. There are several different types of RNA. Messenger RNA, mRNA, is the RNA transcript of RNA copy of the DNA message produced during DNA transcription. Messenger RNA is translated to form proteins. Transfer RNA has a 3-dimensional shape and is necessary for the translation of mRNA in protein synthesis. Ribosomal RNA is a component of ribosomes and is also involved in protein synthesis. RNA most commonly exists as a single-stranded molecule. RNA is composed of a phosphate-ribose sugar backbone and the nitrogenous bases adenine, guanine, cytosine, and uracil. When DNA is transcribed into an RNA transcript during RNA transcription, guanine pairs with cytosine, GC, and adenine pairs with uracil, AU. Differences between DNA and RNA composition. The nucleic acids DNA and RNA differ in composition. The differences are listed as follows. DNA, nitrogenous bases, adenine, guanine, cytosine, and thymine, 5-carbon sugar, deoxyribose. RNA, nitrogenous bases, adenine, guanine, cytosine, and uracil, 5-carbon sugar, ribose. So, main differences between DNA and RNA are first, functional. DNA is genetic information, store of genetic information, and RNA protein synthesis. Second differences is structural. DNA is double chain and RNA is single chain. And the third differences is composition. About this we speak now. So on this picture you can see primary, secondary, tertiary and quaternary structure of nucleic acids. The primary structure have both DNA and RNA. Secondary structure have only DNA, as you understand. And the tertiary and quaternary levels of structure we study in structural levels in proteins. Well, this is a biochemical formula of these biomolecules. Nucleic acid structure refers to structure of nucleic acids such as DNA and RNA. Chemically speaking, DNA and RNA are very similar. Nucleic acid structure is often divided into 4 different levels: primary, secondary, tertiary, and quaternary. Primary, you can see at this picture. Primary structure consists of a linear sequence of nucleotides that are linked together by phosphodiester bonds. It is this linear sequence of nucleotides that make up the primary structure of DNA and RNA. Nucleotide consists of 3 components: nitrogenous base, adenine, guanine, cytosine, thymine, present in DNA only, uracil present in RNA only. 5-carbon sugar, which is called deoxyribose, found in DNA, and ribose, found in RNA. 1 or more phosphate groups. Secondary structure is a set of interactions between bases. The nucleotides on the strange base pair with the nucleotide on the other strange. The secondary structure is responsible for the shape that nucleic acid assumes. The bases in the DNA are classified as purines and pyrimidines. The purines are adenine and guanine. Purines consist of a double ring structure, a 6-membered and a 5-membered ring containing nitrogen. The pyrimidines are cytosine and thymine. It has a single ring structure, a 6-membered ring containing nitrogen. A purine base always pair with a pyrimidine base guanine pairs with cytosine and adenine pairs with thymine or uracil. DNA secondary structure is predominantly determined by base pairing of the two polynucleotide strands wrapped around each other to form a double helix. Although the two strands are aligned by hydrogen bonds in base pair, the stronger forces holding the two strands together are striking interaction between the bases. Tertiary structure you can see on this picture, and quaternary structure is like a nucleotide. So, thank you for your attention. Bye bye.